This is Carroll County's Career and Technology Center's Biomedical Sciences Program Lesson Review. Question 1. What warning signs were there that Dan had skin cancer? While growing up, Dan had a high level of sun exposure. When he was in college, he had a large mole on his back. Over time, it began to increase in size and it would occasionally bleed. When it was removed, the melanoma present in the mole spread to his lymph nodes and eventually the rest of his body. All in all, the mole was a huge warning sign that Dan could possibly have had skin cancer.
Question number 2. How do you think Dan's death could have been prevented? While rowing, Dan did wear a t-shirt. However, the article does not indicate if he wore sunscreen or not. Dan should have worn sunscreen while he was outside for such long periods of time and should have paid more attention to the large mole on his back.
Question 3. When doing a self-examination for suspicious mole or spots on your skin, what should you look for? When performing self-examinations for moles or spots, you should take note of 3 main points: size, color, and shape. You need to make sure that the mole is not becoming larger, that the color is normal, and that it does not have irregular borders or other evolving characteristics. If you have any of these symptoms, make an appointment to see a dermatologist immediately.
Question number 4. Mr. Smith is a landscaper and spends a majority of his time in the sun. What advice would you give him to lower his risk for skin cancer? To lower his risk of cancer, he should wear a sunscreen high in sun protection factor and should wear both clothes that shield his skin and a hat to keep the sun off his face in order to lower his risk for developing skin cancer.
Question 5. Given what you know about these 2 strains of yeast, what role does the cell's ability to repair DNA play and how well it tolerates exposure to sunlight? The wild-type strain is relatively sun-tolerant because it is able to repair its DNA after exposure to sunlight as a result of enzymes. The mutant-type strain does not contain this enzyme for DNA repair and cannot repair damage caused by the UV light. Therefore, a cell's ability to repair DNA is reliant upon having the proper genes that code for this DNA polymerase repair mechanism.
Question 6. Why is it important for a cell to efficiently repair its DNA? It is important for a cell to be able to effectively repair its DNA because proper protein function is dependent upon the correct genetic code. When DNA is exposed to UV light, thymine dimers form in the DNA strand. DNA polymerase II cannot copy DNA that has thymine dimers within the strand. Therefore, they must be cut out and replaced. If the correct DNA sequence is not replaced when the dimers are excised, mutations can occur and a protein can have a defect and have reduced function or become non-functional altogether. If this occurs in a highly conserved protein, this can have deleterious effects on the body.
Question 7. What conclusions can you draw from the experiments you designed and ran? From the experiment you performed, you should conclude that the wild-type yeast strain is sensitive to UV light and SPF 50 protected the yeast from a 15-minute UV exposure. The result of this experiment does not coincide with the expected results. The major issue is the lack of growth on the non-protective side of the plate with the wild-type yeast. This culture should contain growth on both sides of the plate because it contains the enzyme for DNA repair. The mutant strain was expected not to grow on the side exposed to UV light because it lacks the enzyme repairing gene, but was expected to grow on the side that was protected. The dilution ratio did allow for the quantitative results, and it is possible that the exposure time to UV light would need to be reduced by 5 minutes to determine if this is a possible variable. The 15-minute time was set due to the average exposure many individuals experience in a tanning booth.
Question 8. If you were to perform this experiment again, what would you do differently and why? While the data obtained from the experiment are quantitative, I would perform more trials of the experiment so that we could determine if the differences seen between the numbers of UV-protected colonies and non-protected colonies were statistically significant.
Question 9. If yeast cells repair damage to their DNA in a similar manner to humans, what do these results tell you about the degree of DNA repair that occurs in our cells each time we expose ourselves to sunlight? These observations suggest the DNA repair mechanisms are highly conserved across species and have been an exceptionally important evolutionary adaptation that has allowed us to thrive on Earth despite a constant exposure of UV light. For example, like the wild-type yeast exposure to UV light. But we must protect against the eventual damage caused by UV that is very similar to winning the lottery, a very bad one, with the grand prize of skin cancer.
Question 10. You have been contracted to design a new medical intervention to help prevent skin cancer. What type of medical intervention would you design? Describe your medical intervention and explain how it works to prevent skin cancer. I would design a sunscreen that contains a synthetic melanin stimulation hormone that triggers melanocytes to temporarily increase melanin production. In addition to the protection conveyed from the sunscreen, the increased melanin production would increase UV protection and ameliorate some of the negative effects of UV rays.
This concludes activity 3.2.2.